Hi, everyone, and good evening. Folks are still rolling on in. I'm so happy you guys found your way here with us tonight. All right. Let's get started. So my name's Allie Gustafson, your community organizer with KS Wild, and I'm calling in from the unceded ancestral lands of the native tribes of the Tequilma people that is now called Grants Pass, Oregon. They are not only important parts of the history of this place, but in the future and the continuing knowledge of this place. So if you don't know whose land you're on right now, go find out. Educate yourself with the place you live and the work that those tribal nations are advocating for. Other ways you can support tribal peoples is to make a donation to your local tribal nations. And a good place to start is checking out the Indigenous resource page on the KS Wild website. I'll put that link in the chat box soon, so keep a lookout for it. It's also fun, since I can't see any of your faces here tonight, to see where you are all calling in from. So for those of you who are with us right now, navigate to the chat box and let us know where you're zooming in from tonight. I'm so happy you are with us tonight. For those of you who are here, we have a great presentation in store for you titled, Why Serpentine Rocks? This is part 1 of 2, titled Serpentine Plant-Soil Relations, A Worldview. But before we begin, I have a few announcements. The Love Where You Live webinar series is produced by Klamath Wildland Center. We are an environmental nonprofit working to restore wild nature in the Klamath-Siskiyou region. We promote science-based land and water conservation through policy and community action, which is all of you. So thank you for being with us tonight and for your support to protect the Klamath-Siskiyou ecoregion. With that, I want to let everyone know that this session will be recorded, and we've saved time at the end for a Q&A session. Now, get comfy, sit back with your favorite beverage, and I hope you enjoy tonight's presentation. And now, I'd like to introduce our guest speaker, Nishi Rajakaruna, author, researcher, and is a professor in the science department at Cal Poly State University. Nishi fell in love with serpentine and all things geobotanical as an undergraduate student. Since then, he has done research on diversity, ecology, and the evolution of plants, lichens, and microbes on serpentine and other harsh soils here in California, here in the Siskiyous, here in Southern Oregon, where he spent time researching, in Eastern North America, South Africa, Sri Lanka, where he's from, and Russia. As a member of the International Society of Serpentine Ecology, he has botanized on serpentine outcrops across the world, and along with our longtime friend and advocate, Susan Harrison. They both co-edited a book titled Serpentine, Evolution and Ecology of a Model System. Thank you for being with us tonight, Nishi. I'll now hand it over to you.
Thank you so much, Ali, for that kind introduction. Great to be with you guys. I wish we could do this in person. Of course, we are unable to do that, but hopefully in the coming year, I can get up there and actually get out in the field with you and talk to you a little bit about serpentine plants and other critters that grow in serpentine habitats. So I'm going to share my screen with you. So thank you also for making time to attend my seminar. I know like we are all going through Zoom fatigue. It's not the most exciting thing to do on a Thursday evening. So I appreciate you showing up to listen to my talk this evening. Thank you. And also, if there are any questions along the way, I don't know whether you are able to ask those questions because this is a webinar, but if there is a way to ask those questions, please reach out to me or put it in the chat, but I'm not sure whether I will see the chat as I go along, but we'll try to get to your questions at the end of the seminar. Ali, is that good? You either let me know if there's a problem, if there are questions, or we
Yes, Nishi, I'll let you know. And yes,
cool.
All the attendees can type their questions in the chat box or the Q&A box. Thanks,
Perfect.
Nishi, for clarifying that.
All right, thank you so much. All right, guys. So I've been fascinated with serpentine and all things geobotanical, like Ali said, for a long time. I love hanging out on rock outcrops. I like contaminated mine tailings. I like all sorts of places where rocks shape the diversity around where we are. So today's talk is going to be kind of an overview of plant, lichen, microbe associations with serpentine habitats, both here in California as well as in other parts of the world. So when Ali invited me for this talk, I was thrilled because I've never had a webinar series named after my bumper sticker on my car, which basically says Serpentine Rocks. So when I had a webinar series, when you guys invited me to give a talk on a webinar series called Serpentine Rocks, I just couldn't say no. So I'm honored to be a part of this webinar series. So thank you for the invitation. So my fascination with serpentine, like Ali pointed out, actually started when I was an undergrad, maybe third year, fourth year. I took a plant systematics class at College of the Atlantic in Maine, and I came across these two papers during a plant systematics class. And those papers, I read them, I reread them, and I was hooked. I was fascinated with serpentine. I was fascinated with how geology and landforms shape diversity. And, you know, this all happened even before I had stepped on a serpentine outcrop. I've never seen serpentine in my life, but these writings of Art Kruckeberg that some of you may have met, taken classes with, or have heard of, basically kind of got me hooked in this field of serpentine geoecology or serpentine botany. Art has had a significant impact on Western North American geobotany, particularly serpentine ecology. He's written a bunch of books. He's written about 30 peer-reviewed publications that have been cited very heavily over the years. And then on top of that, he has been really a kind of a key figure in promoting geobotany as a subject, as a discipline across this country, as well as in other parts of the world. He's a highly esteemed figure in the international community of serpentine ecologists all over the world. Everybody knows who Art Kruckeberg is. And I had the pleasure of first meeting him at the Siskiyou Field Institute in 1999. And I was just telling Ali that I was asked to come and be a teaching assistant for him in 1999, and I didn't know anything about the Klamath Siskiyou region. And very quickly, I realized that all Art wanted me to do was to drive him to all of the nearby wineries at the end of the day. So I was the designated driver, AKA teaching assistant, for the course that Art Kruckeberg and Bob Coleman taught at the Siskiyou Field Institute. So I was also honored when people asked me to write his in memoriam when Art passed on in 2016. You can find this in Madronio, which is the Western North American Journal of Botany. I've got the citation over there where I explain my relationship to Art and how Art's writing and my relationship with him since I first met him shaped my career over the years. So in addition to having Art as somebody who introduced me to where you guys live, at least most of you live, I've also read the Klamath Knot. I've reread it. I've given it to my students to read, and I just love that book for the amazing work it does in terms of introducing your region to the world, the natural history of the area. And at the same time, I do have a poster from, I think it's from the Oregon Native Plant Society in my office that reminds me of the Klamath Siskiyou region every day I walk into the office. And of course, I have memories and photos in an album, which I didn't have time to scan from my time at the Siskiyou Field Institute in the late 90s. And then, of course, I've got to show you the T-shirt that Susan Harrison sent me just last year of the Klamath Siskiyou area, the Siskiyou native plants that grow on serpentine in the Siskiyou region. So how can you not love where you guys live? You guys live in an amazing serpentine country. And, you know, I wish I was closer to where you guys live because I just love botanizing in that area. And I'm sure you're going to get a real nice treat from John Roth, who I think I met in 1999 when he gives the second part of this series in August. So briefly, I know I'm a professor at Cal Poly. I've been here since 2017. Prior to that, I taught on the East Coast at a place called College of the Atlantic. And most of my research is on serpentine, but I'm also interested in other harsh soil types like gabbro, metal mines, chromite mines, even bird nesting sites where the guano can be a pretty harsh substrate for plants to grow in. Recently, I've started working in Carrizo Plain National Monument, where there's vertic clay soils. These are soils that have over 50% clay and really high sodium content. And no one actually has really studied how plants adapt to these vertic clay soils. So I, along with a grad student and a collaborator, Ryan Odell, who you may have met or heard of from the BLM, are studying plant adaptation to vertic clay soils. I'm also interested in plants that grow on vernal pools, as well as in carbonate-rich soils like limestone, gypsum, and dolomite. I also teach classes focusing on plant communities that grow on these harsh soils. When I was at College of the Atlantic, I taught a class called Plants with Metal, these courageous little plants that live in metal-rich soils. And I was working mostly at a copper zinc mine, which was near the school. And then, of course, at Cal Poly, I've started a new class called Geoecology, an upper division ecology class, looking at how landforms and lithology, parent material, shape diversity around us. So what do I mean by a harsh soil, right? I mean, so we're talking about serpentine, but I wanted to kind of take a step back and introduce you to what I refer to as a harsh substrate, harsh for the plants, right? So any kind of soil that has too much or too little of something that plants require for growth can be a harsh substrate. Too much of something can lead to toxicity. Even something like nitrogen and phosphorus, too much of it can be toxic, and too little of it can lead to a deficiency, right? So plants have a hard time, physiologically speaking, dealing with this kind of a substrate. There are some soils, like serpentine soils, that have toxic elements that plants don't require for growth, right? So think about serpentine, where most of our nickel and chromium comes from, cadmium, cobalt, all these heavy metals that plants don't require for growth, but it's in the soil. So plants have to be able to deal with that toxicity. And if those elements are in the soil, that can also cause a level of harshness in terms of the physiological response of the plant to that substrate. In addition to that, harsh soils can be soils that have poor structure, shallow soils, soils that have too much sand, too much rock, or too much clay, where the roots have a hard time growing or extracting water and nutrients from, and that can also contribute to what I refer to as harsh. So all of these factors are part of serpentine. All of these factors are part of soils like limestone, vertic clays, guano soils, and these are the kinds of substrates that I use as a model system to ask questions about diversity, ecology, evolution, and at the same time, about questions about how to conserve plants that grow on these kinds of sites. How do you restore landscapes that are disturbed on these types of soils? So those are the kinds of questions that I'm interested in and have been pursuing over the last 20 plus years. So when plants are exposed to harsh substrates, like the one you see in this picture, which is from where I come from. I'm from Sri Lanka, and this is one of our southernmost serpentine outcrops overlooking the Indian Ocean, a beautiful serpentine setting. And what you see is a serpentine, non-serpentine boundary. There's more kind of sandstony soil to the left on the top of the picture, and then you have serpentinite, mostly kind of hematite-rich, iron-rich, clay serpentine soil. Very few things grow here. The ones that are growing on the serpentine outcrops are mostly prostrate herbs. There are some interesting plants that are on this outcrop that are not found elsewhere. And so it's an interesting setting, but this kind of a sharp vegetation boundary between serpentine and non-serpentine soil is found all over the world, including in the Klamath Siskiyou region where you guys are from. So when plants encounter harsh soil like this, either if they're not tolerant, they will be excluded from that substrate. They will not just be able to tolerate it and live on this substrate. So these are called intolerant species, serpentine intolerant species. and they are excluded from these substrates. Then there are plants that are found on and off. I mean, we find these plants all over California. You find them on serpentine soil, you find them off serpentine soil. So these are the ones that have broad tolerance, broad substrate tolerance, or they may even have ecotypes, specialized populations that look the same on and off of serpentine. So these are the kinds of ones that Kruckeberg called bodenwogs, or soil wanderers, right? They seem to be indifferent to the substrate chemistry and the substrate conditions. Then, of course, we have our special group that we all in California love, the serpentine endemics, the ones that are largely restricted to serpentine. So these are what we call the serpentine endemic species. These are plants that are able to tolerate the harsh conditions on serpentine soil, and they are so specialized that they get outcompeted from non-serpentine settings, and they are restricted to serpentine soil. And in California, we have about 277 species that are restricted to serpentine soil. So geology, broadly speaking, is really important in terms of contributing to habitat diversity and discontinuity, right? If you think about climate, it's really important, right? We know that if you go to the northern parts of the hemisphere, you get a different type of vegetation than you get when you get to the southern part of the equatorial regions, right? But even within a climatic regime, under the same climatic envelope, geology contributes to diversity and discontinuity of species. So here's a great example from Stanford's Jasper Ridge Biological Preserve, where I worked as a grad student. So what you find here up on the top part of the slide is the serpentinite outcrop. Nutrient-poor soils, heavy metals are high, and then it is adjacent to a sandstone outcrop, right? So the serpentine outcrop and the sandstone outcrop has a very sharp boundary that you see from this image over here, where you have serpentinite on the top and the sandstone on the bottom. And these kinds of sharp geologic boundaries are found all across California. So what you find is that the serpentine outcrop is dominated in this particular case by a species called Lastenia californica, which is a California common goldfield. And then on the sandstone outcrop, you have native and non-native grasses, and a lot of non-native grasses at this particular site. So when you do reciprocal transplants, right, to understand whether the geology is actually contributing to this sharp boundary pattern that appears year after year after year, you move the yellow plants to the sandstone, you move the grass to the serpentine, and you see how they perform. When you do that, you realize that the serpentine, the grasses, especially the non-native grasses, don't do so well on the serpentine soil. And then the serpentine endemics, or the serpentine tolerant species, get outcompeted in the sandstone because they are stress tolerant species that are unable to kind of grow as fast as the grasses do on the sandstone. So they get outshaded and they kicked out. So both abiotic stress, soil chemical stressors, and the biotic competition component contribute to maintaining this really sharp boundary across this geologic boundary. So next time you are out there, you see these boundaries, realize that there is this kind of active, war going on, or, competition and selection happening across these boundaries to show the patterns of diversity you see on these rocks that are in front of you. So I think about the world as a patchwork quilt, right? There's all these patches of soil types, geologies, that in each of those patches, you might get one or a few species or a community that is better able to deal with these conditions than another plant or another community. So over time, what you get is you get these distinct substrates that become homes or sites for locally adapted plant populations. And over time, these plant populations generate plant communities. And that's where you get these really sharply defined geologic boundaries that are occupied or colonized by these different communities that look different across those geologic boundaries. Right? So if you think about California, 50% of California's endemic species, that's about 1,240 species, are substrate specialists, meaning that they are either on serpentine, they're on granite, they're on sandstone, they're on limestone type substrates, they're on shale, vertic clays. So all of these substrates that you have across California are home to specialized species that also happen to be California endemics, right? So substrate plays a huge role in generating California's plant diversity. And interestingly, half of these plants that are substrate specialist and California endemics are also rare plants. They're listed by the California Native Plant Society and the rare plant ranks as rare, right? So half of the California endemics that happen to be substrate specialists are rare, which really shows that if you want to think about conserving California's plant diversity and restoring areas that have been disturbed, you've really got to pay attention to the substrate plant association. Unless we know the relationship that the plant has with the soil, conservation efforts and restoration efforts are not going to succeed, right? So this is an important take-home point to realize that we have to start paying more attention to studying the below ground relationships plants have with their substrate if we want to think about conservation, especially in light of climate change and all of these anthropogenic stressors that plants are dealing with right now. A really important point that many of California's endemic species are substrate specialists, meaning that they're endemic to one type of substrate. And then many of them, at least half of them, are also listed as rare across California, which is a huge point of concern. So while the talk is on serpentine, I also want to point out there are a lot of these underappreciated, understudied substrates. I want people to go out in an army and explore, because like serpentine is special, there are these other substrates that also have endemic, highly restricted or largely restricted species that require more study. Right? So I'm just showing you a few of these substrates that we know have specialized plants that require additional study. While we keep studying serpentine, we want to take a step back and explore other types of geologies that are really found across California and Oregon. So for example, now that I'm in San Luis Obispo, very close to Carrizo Plain National Monument, which is famous for the super bloom, right, that people go to when good rain years generate these massive displays of color across the Carrizo Plain National Monument. When I came over here, I realized that there weren't that many studies done on understanding how plants adapt to these highly clayey soils of the Carrizo Plain, right? So these soils have over 50% clay. They have really high shrink swell capacity, which makes it really hard for the roots to grow in. In addition to that, these soils have gypsum, which is calcium sulfate, sodium, selenium in the soil, which can also be toxic to plants. So how do plants adapt to these types of soils, right? Is something that my grad student is studying using these endemic species that are largely restricted to these vertic clay soils that have different degrees of salt and clay in those places where they grow. So Emma Fryer, who actually came from Humboldt State University, where she did an undergrad, is now studying with me here at Cal Poly, trying to understand how these vertic clay communities come together. What are the stressors that these plants experience? How do they compete with the invasive grass called Bromus madritensis that seems to be encroaching these vertic clay soils? So we're doing a greenhouse study looking at competition with Bromus madritensis as well as adaptation to varying degrees of salt and clay that is found in the Carrizo Plain National Monument. So I don't know, Ali, whether there are any questions from the introduction yet. I can quickly go through that. All right, doesn't seem like there's any real questions. Great to see that you guys are from Ashland and all these cool places up north. All right, so serpentine is the kind of the big substrate that we have all fallen in love with. Everybody's studying it. And my pitch earlier was to say, okay, while we're studying this, we should take a step back and look at other substrates that are out there that are equally exciting as serpentine is, right? Lots of books have appeared, including the book that Ali just talked about from 2011, Serpentine Model, Evolution and Ecology in a Model System, which Susan Harrison and I co-edited. So really a lot of work done, a lot of people interested in serpentine relative compared to other types of substrates that I've just talked about. So what are serpentines? These are basically mantle rocks that kind of come up in places where there are subduction zones or continental margins, places where there's orogenic activity, mountain building activity, chunks of the mantle can get popped up. There are mid-oceanic ridges that have serpentinite substrates in them. These are the soils that weather from this type of rock end up becoming called serpentine soil, right? So serpentine is just a kind of a loosely used word we use to describe any type of soil that has weathered from this kind of ultramafic, high iron, magnesium rich serpentinite type rocks, right? So there's a whole suite of rocks that are called ultramafic, and the soils that are weathered from this are called serpentine soils. They have high heavy metals. They have nutrient imbalances. Nutrients are generally low, not everywhere in the world, but at least in California, especially in kind of chaparral settings, you find that the nitrogen and the phosphorus levels are pretty low. The calcium to magnesium ratio is really important. Plants require both calcium and magnesium, but more calcium than magnesium. But in serpentinite soils, the magnesium is elevated and calcium is low. And that causes physiological stress. In addition to that, there are open habitats. Many places that you see serpentine, they're pretty exposed to the elements, right? It's hot, shallow soils. It dries up pretty quickly. So plants are exposed to drought stress often, especially in Mediterranean climate areas where there's serpentine soils. But all of these stressors contribute to these distinct plant communities that we have all fallen in love with, that have high levels of endemism and many, many species that are incredibly rare. Right. So this is why serpentinite is so important in California and very aptly named as California State Rock. Serpentines are found everywhere, right? I mean, this talk is about the world view. This is found everywhere, including the East Coast and the West Coast, and all of these little blue dots that you see, and the triangles that you see are either kind of ultramafic rocks or sedimentary type serpentinite settings. So there, about 1% of the Earth's surface is covered by exposed serpentinite rocks where plants have an opportunity to colonize. Serpentinite are important in our lives. Asbestos comes from serpentine. Stainless steel comes from serpentine, right? And then if you go to places like Cuba, South Africa, parts of the southern Urals of Russia, you have people selling jewelry and little sculptures made out of serpentinite. It's easy to work with. Right? And there are in places like Portugal and Italy and Spain, you have churches that are made out of serpentinite rock, right? So serpentinite have been important even in terms of human culture, where people have used these rocks, just like they have used soapstone to carve things. And I love running into these things. In fact, I have a collection of little things that I picked up from around the world where people have carved out of serpentinite or other type of ultramafic rock. California is cool in terms of serpentine endemism, but there are other places that are even cooler in terms of serpentine endemism. New Caledonia, these islands off the northeast of Australia, are probably the biggest hotspot for serpentine endemism. 83% of New Caledonian endemics are restricted to serpentine soils. Cuba is another fascinating set of islands where you find a lot of endemic species that are restricted to serpentine soil, close to 1,000 species. Sabah, Malaysia, recently being studied by my colleague Anthony van der End from the University of Queensland. They're finding endemic species every trip they make to these serpentine outcrops in Malaysia. And they think that about 5% of the total vascular flora of Malaysia is endemic to serpentine soils. And most of them are restricted to this province of Sabah, where Mount Kinabalu is. Right? So a quick place to show you where New Caledonia is, just off the east of North Australia. Lots and different types of habitat. There are rainforests on serpentine outcrops in New Caledonia. There are chaparral type settings. There are grasslands, a lot of different communities. And there are 20 genera that have just one species in them, so monospecific genera, and 8 families that are endemic to serpentine soil. Nowhere in the world do you get an entire family restricted to serpentine soil. And in New Caledonia, you get 8 families. In Cuba, where I had the great fortune of being with Art Kruckeberg on the left, Richard Coleman, Dick Coleman from University of Washington, who was the first person to study plant adaptation to calcium-magnesium ratio on serpentine soil. Bob Coleman, the Stanford geologist who used to teach with Art at the Siskiyou Field Institute, and Earl Alexander, who is the only person living among these 4 giants here, who's a pedologist, a soil scientist, who who has also spent a lot of time in the Klamath region. So this is like one of the times the only time I actually managed to be in the field with all these giants of Western North American serpentine geoecology. And in Cuba, there are over 900 species, now close to 1,000 restricted to serpentine soil. I also had the great fortune of being on Mount Kinabalu in Sabah, Malaysia in 2014, where there are about 350 species endemic to serpentine soil. Mount Kinabalu is the highest peak in Southeast Asia, and some of it is serpentinite rock. And on these serpentinite settings, you have orchids that are endemic to serpentine soils. These photos were taken by my colleague, Anthony van der End, but these are beautiful orchids that are restricted to serpentinite soils on Mount Kinabalu region. There are pitcher plants, right? These are massive pitcher plants. I mean, we have our cobra lily, like the Darlingtonia, in Mount Kinabalu. There are a number of pitcher plants in the genus Nepenthes that are restricted to serpentinite soils, which makes sense, right? I mean, serpentine soils are nutrient poor, and these kinds of areas are perfect settings for insectivorous habit to evolve, where plants can trap insects to basically supplement their nutritional needs. So in terms of California, right, I mean, going back to California, about 1.5% of the state has serpentinite, 15% or so of California endemics are serpentine endemics. And of course, you know, some of the ones that you guys know are largely restricted to serpentine, like the Darlingtonia californias that I'm sure you guys have seen in the field. And then of course, the California lady slipper, the Cypripedium californicum, which are largely restricted to serpentine fens up in the northern parts of California and southern Oregon. And these are some of the charismatic species you come across when you look at those 255 taxa that are largely restricted to serpentine soil in California. Where I live, there are many species that are named after San Luis Obispo because the plants are largely restricted to San Luis Obispo County and were collected, initially named from specimens that were collected here. So the Calocotus obispoensis, which was in flower up until about 2-3 weeks ago, is found largely in San Luis Obispo. The southernmost range of the southern cypress is in San Luis Obispo. It goes a little further south. There are carex species, there are dudleyas, there are bog thistles, and there are manzanitas that are restricted to serpentine and named as obispoensis where I am. So when I got this job, or when I heard about this job, I wanted to get here because other than being at a place like Klamath Siskiyou, this is not a bad place to be. There are a lot of serpentine endemics out here as well. In fact, my colleague and our senior botanist, who's now retired, Dave Keel, named a new serpentine endemic plant in the genus Corysanthe about 2 years ago in 2019, called Corysanthe aphanantha, which actually grows with another Corysanthe called Corysanthe breweri, right? So they are sometimes sympatric, meaning they grow together. Sometimes they're separated, but we have been studying their ecology, and we're finding that the aphanantha, which is known as the Irish Hills spine flower, which is because it was identified and is only found in the Irish Hills region of San Luis Obispo, is found in serpentine soils that are generally higher in organic matter content, higher in cation exchange capacity, and lower pH. So there's subtle differences in the soils where these close relatives are growing. And we're studying whether there's reproductive compatibility. We're studying their chromosome numbers to really understand what the relationship is between these Corysanthe species that are growing together at some of the serpentine outcrops in San Luis Obispo. There was a little monkey flower that was just described last year by also Dave Keel and colleagues called Erythranthe guttata. And this is a little serpentine monkey flower that is also found in Irish Hills area. And there's a grad student here studying this particular plant and finding that they're much, much smaller than your typical Erythranthe guttata, the monkey flower, the seep monkey flower. And this guy generally grows away from the seep, and they're generally coming right off the little cracks of the rock. So it seems to be growing right off the rock. And it seems to have an annual growth habit compared to Mimulus guttata or Erythranthe guttata, the seep monkey flower we all know of, that is mostly a perennial. So there are subtle differences in terms of the life history of this plant, which was recently described as a new species as well from San Luis Obispo. If you look at California, across California, what you find is that plants that are found, the northern serpentinites of California, where you are from, have more endemics than the ones in the south, right? And part of the reason that Susan Harrison and colleagues have found out is that higher precipitation leads to greater diversity, greater restriction into harsh settings, larger areas of serpentinite outcrops. Klamath region has the largest serpentinite area in North America, so larger areas generate greater habitat, and also the longer exposure of rock up in the north may have contributed to kind of greater, longer time for evolution to take place. So therefore, the northern parts of the California and the North Coast ranges have higher levels of endemism than places in the southern parts of California. So people in California don't realize that there's serpentine on the East Coast, right? There's almost as much exposed, not exposed, serpentine or serpentinites on the East Coast as you get in California. The 600,000 hectares, which is about what we get on the West Coast, all the way along the Appalachian Belt, from Newfoundland down to Georgia and Alabama. And the tiny outcrops, many of them are under glacial till, some of them are exposed. And when I was on the East Coast, we studied some of the serpentine plants of the eastern outcrops. There are at least 6 serpentine endemics on the East Coast, nothing like the 255 we have here in California, but there are endemics in Eastern North America. I mean, they're not as abundant because it's colder climate, right? And a shorter growing season. These rock outcrops are under glacial till. So there hasn't been a lot of time for evolution to take place because these areas were under ice 10,000 years ago, right? It's not like a lot of ice-free time that you get here on the West Coast, but there are serpentine endemics, including this Pachyra species that was the most recently described serpentine endemic from Eastern North America. So these non-serpentine serpentine boundaries have been model settings to study evolution. How do new species originate, right? So these boundaries are perfect settings to study natural selection in action, because you have these 2 types of substrates that have very different from each other. That can create stress, that can then generate these serpentine tolerant genotypes or select for genotypes, genetic variants within a population that is able to deal with this new set of conditions. Right? And over time, these plants could potentially become their own species. So these boundaries are model settings to understand the evolutionary process. A lot of work has gone into this, and serpentine outcrops are often used as a model to talk about rapid evolution or evolution under the influence of geology or soil conditions. In fact, what you see here is 2 lineages of Arabidopsis species from Europe, where people are basically growing them on serpentine soils and non-serpentine soils. So what you get is serpentine origin population growing on non-serpentine soil and serpentine soil, and then a population of the same species or the same lineage growing on non-serpentine soil and serpentine soil. So what you find is that when you grow the serpentine origin plants on serpentine or non-serpentine, they do fine, right? They are able to deal with both serpentine and non-serpentine, especially when they're growing alone, right? When they don't have competition from non-serpentine plants. But when you grow the non-serpentine population on serpentine soil, they don't do well. They just don't have the adaptations that they need to tolerate and thrive on serpentine soil. So there's generally a reduction in growth or sometimes outright mortality, right? So this is kind of understanding how adaptation could potentially eventually drive evolution. So there are all of these cool experiments that people have done looking at populations on and off of serpentine soil and shown that they're pretty distinct in terms of their ability to deal with soil. So plants see soil, right? They understand soil, and they have to because they're rooted, right? They're rooted in soil. And that's why it kind of sometimes frustrates me that botanists don't pay attention to the plant-soil interface, because that is so critical, as more critical than perhaps even thinking about the above ground factors that plants have to deal with. So a lot of genetic work has gone on to understand serpentine adaptation. We know that there are many genes involved in terms of serpentine tolerance. So it's a multi-trait system of adaptation. Calcium, magnesium, iron tolerance seems to be really critical across all of these model genetic studies that are done on model systems like the monkey flowers and the Arabidopsis and the Silene species, where people in Europe and America have studied the genetic basis for tolerance to serpentine. Heavy metal detoxification, how do you deal with nickel? How do you deal with chromium? How do you deal with cadmium that are toxic? Also seem to be important between serpentine tolerant populations and intolerant populations, between the two populations that are either tolerant or intolerant. Drought tolerance seems to be really important as well, because as you know, many serpentine outcrops are incredibly dry. They dry up quickly, they're shallow soils, very little water holding capacity. So plants have to deal with water stress. So these are some of the important features with respect to understanding adaptation to serpentine soil. Plants have to undergo all these genetic changes over time to be able to live and thrive on these types of settings. There are also studies that have looked at how serpentine plants are isolated from their closest relatives that are found off serpentine soil. Because plants originate from various populations, right? Genotypes originate, but if the genotypes continue to interbreed with each other, then there won't be a new species that could potentially form, because they're constantly mixing genes. But what you find is that in many cases, when you have new populations that colonize serpentine soil or other types of harsh soils like mine tailings, right? What you find is that there are flowering time differences that also evolve, that then immediately cut off potential gene flow between these sets of populations. There are switches in their breeding system. Sometimes serpentine endemic populations can self, meaning that they can use their own pollen to make seed, which means that they don't need to get pollen from another populations. That can also cut off potential gene flow. There are hybrid sterility issues, hybrid fitness issues between serpentine tolerant populations and intolerant populations. There are also pollinator shifts. Serpentine plants, like the little mimulus you saw, have a tiny flower compared to their presumed ancestor that has a bigger flower. Right? So the pollinator, the bumblebee that pollinated the bigger flower cannot go into this tiny flower, right? So therefore, you end up immediately cutting off potential pollination between the ancestral population and the derived population. So unless if a native bee or another insect picks up pollinating these little flowers, then this potential population can start interbreeding within each other, within the population, which could then lead to the origin of a new species. So pollinator shifts have also been important between populations on and off of serpentine soils. There's this cool set of studies coming from Pennsylvania, looking at Streptanthus glandulosus, Streptanthus polygloides, which is found in the Sierras of California, where they find that the heavy metals accumulate in the nectaries or the nectar of Streptanthus. And the heavy metals are toxic to some of the pollinators. So some of the pollinators don't visit plants that are growing on serpentine soil because the nectar is enriched with heavy metals. So only heavy metal tolerant insects will pollinate serpentine populations, thereby also cutting off potential gene flow between these populations, right? So the big picture here is that serpentine plants have a really unique genetics, often that are different from their closest relatives. And the second point that I want to make is that these populations that evolve, mutations happen all the time. And when these populations evolve or individuals arise with these mutations, if they become isolated reproductively, there's potential for these things to become new species, right? So this is happening as we speak, right? Some make it all the way to a California endemic, some don't, right? And it's important to think about some of these processes that drive this evolutionary process. So even at the Jasper Ridge site that I pointed out earlier, there are two separate species that grow side by side on the serpentine outcrop, right? This is diversification or separation on a serpentine soil based on little elevational gradient, where you have wetter, high sodium magnesium soils at the bottom of the serpentine outcrop because of the elevational gradient topography, and then the upper reaches of the serpentine soil is dry and sandy, right? So this boundary between the two is maintained year after year after year, because even though they germinate across the outcrop, by the time they flower, selection has weeded out the unfit types. You have Lecinia gracilis up on the top and Lecinia californica at the bottom. When we did various greenhouse studies and field studies, we saw that the one is more drought tolerant than the other one is more salt tolerant, right? But requires more water. And then when you do reciprocal transplants, move them up and down, we find that they're not good at dealing with each other's habitat, but they grow side by side, right? From here to here is only about 40 meters. And then Lecinia californica starts around the 30th meter, right? But they maintain this boundary year after year after year because they have different adaptations to deal with these types of soils. They're cryptic species. They look alike, more or less, right? But they know what type of soil they grow on. We've also done studies to show that there's very limited gene flow between these populations because they flower at different times. There's reduced compatibility between the two as well. All right, so I've got another, let's see, I've got another 10 minutes or so. I'm going to quickly transition into other types of things that are important on serpentine soils. I was lucky enough to get to South Africa a few times, and we were granted a National Geographic Society expedition grant recently to go to South Africa to study lichens. The last time lichens have been studied in South Africa was in 1957. So we were interested in looking at lichen diversity on serpentinite rocks and sandstone and other types of rocks, sedimentary rocks that were side by side, right? And we were looking at the influence of both climate and geology to lichen diversity in South Africa. And our hypothesis was that in areas where there's a lot of rainfall, like in the Klamath Mountains, you would find more endemic lichens or lichens that are largely restricted to serpentinite rocks. So when we went there, we realized that none of the lichenologists I took with me from North America knew anything about South African lichens. I mean, we would call them orange blob or black, blob, or we just name things because nobody knew what they were. We collected over 1,000 specimens. We have so far identified only about 100 specimens, 2 species, using mostly genetic tools. And we now have about 6 to 8 species that we think are new to science. And we just recently described 1 genus new to science. And this all happens over a 2-week field trip, right? The problem now is that we have all these specimens that we don't know what the heck they are. We're sending them all around the world, try to identify what they are, right? So the genus that we described was recently published in the South African Journal of Botany. This is my former undergraduate student, Ian Medeiros, who's now a PhD student at Duke, studying lichenology. And we got a bit of coverage for describing a new genus of lichen from South Africa. And what I did was transfer this study to California, and my grad student, Michael Mulroy, I don't have time to go through this video, but he's studying the same thing here in the Central Coast, where he's basically looking at coastal inland gradient of lichen diversity on serpentinite and sandstone soil rocks. These are saxicolous lichens that are found only on serpentine, on rocks. And we are comparing diversity along this coast inland gradient, where there's salt on the coast, no salt inside, and there of limited salt in the air on the inland sites. And we have found a lot of interesting species that Michael is working through right now. If we have time at the end, I'll show this little video that Michael put together. All right, so in addition to lichens, we've also been studying biological soil crust in South Africa. And these are little crust-like prokaryotes, cyanoprokaryotes, blue-green algae that are found on top of soil. And we've described 9 genera that have never been reported for South Africa ever, found on serpentine soils across the region that we study. So there's a lot of work to be done beyond vascular plants. Mycorrhizae, fungi that grow in serpentine soils, play an important role in plant tolerance to serpentine soil. There are some studies on mycorrhizae, but we need more. Bacteria, biological soil crust, plant-insect interactions. These are all important things to look at in terms of serpentine soil. So the last thing I'm going to talk about is this really cool applied green technology that has risen from studying serpentine plants. Right? This is called phytomining that I'm going to get to in the next 10 minutes we have. So there are plants on serpentine soil that can take up huge amounts of heavy metals into leaf tissue. These are like vacuum cleaners that suck up toxic metals all the way into the leaf, right? And people first discovered this in the 80s. They were talking about it in the 90s. This appeared on BBC and CNN, New York Times. People started getting excited about it. Now there's a lot of research going on in terms of understanding how serpentine plants, some of them, can take up really, really high levels of heavy metals into their leaf tissue. The cutoff to call a hyperaccumulator is 1 milligram of metal per gram dry leaf tissue. Some of them take up way more than that, right? And you'll hear about some of them from various parts of the world. So the first kind of indication of plant tolerance to nickel came from a diary of an Italian naturalist from the 1500s, 1583. He said that if you find this particular plant, which is in the Brassicaceae, on a particular outcrop, that has good enough nickel to mine. So he was using this plant as an indicator for mining, for prospecting for metal in rocks, right? And sure enough, this is a serpentine endemic plant that takes up really high levels of nickel into the leaf tissue, right? Back in the day, we didn't know any of that, but he made the association that this plant is restricted to serpentinite rocks. And now we know it's not only restricted, it is also what we call one of the metal hyperaccumulators on serpentine soil. It has about 1 to 2% nickel in above ground tissue. Nickel is toxic. 1 to 2% nickel in our body could kill us, right? Or could seriously harm a young child. But plants are able to, some plants on serpentine soils can deal with this. The biggest discovery of a serpentine associated nickel hyperaccumulator happened in the late 70s, where they found a small tree in the Serpentaceae in New Caledonia that had over 25% dry weight of nickel in the latex. Like, this is bleeding nickel out of the plant, right? Every part of the plant had more than that 0.1% cutoff, or the 1 ppm, 1 milligram cutoff, right? So this was the kind of the king of all nickel hyperaccumulators. And here's Anthony van der End. We might run over by a couple minutes. I hope that's okay. I want to show you this little tiny video clip of Anthony looking at a plant in New Caledonia that is a nickel hyperaccumulator.
Here in the jungle of Grande Terre, scientist Anthony van der End of the Australian University of Queensland is researching plants with bizarre abilities. Plants that can simply draw heavy metals such as nickel from the ground.
We have a really large individual of Pichinendra cutuminata here, which is famous all around the world for containing a blue-green sap that has 25% nickel in it. So I'm going to make a small cut in the tree to exudate some sap that we then can collect and analyze. We should see some sap coming out soon.
Basically the latex that he's referring to.
The green color you can see from the sap is actually the color of nickel. I can use a little bit of test paper to press against it, and we should see a pretty strong color reaction. So the amount of nickel in this sap is between 20 and 25%, which makes it the strangest biological liquid known to man. That is really quite extraordinary to think there's so much metal contained in the sap of a living, healthy tree.
All right, so that's a quick kind of a view of Anthony cutting a little chunk of the Serbetia from Pichinendra from New Caledonia. So they've found a lot of plants all across the world where not only does the latex have nickel, even the phloem tissue, out innermost part of the bark, which is phloem that transfers sugar, has huge amounts of nickel, including this Phyllanthus species from Malaysia that they found 17% nickel by dry weight in the phloem sap. This type of level of nickel could kill any big organism, and the plants are putting them in sensitive tissue like the phloem, which is what they use to transport sugar. Right? They also put them in the leaves. They put them in the little leaf hairs on the trichomes. Right? This is a image of a plant where nickel is enriched on the upper epidermis and the lower epidermis. Right? So the skin of the plant is also enriched with nickel. And there are test methods, field test methods. If you guys are interested, I can send you the little recipe to make little field filter paper you can carry with you when you're traveling in the Klamaths, crushing leaves, and if it turns bluish green, you know that you have at least 1% nickel in the leaf tissue. Very reliable field test, and I can talk to you about it if you're interested later. Why do plants do this? Why do you put a toxic heavy metal in sensitive tissue? The idea is that it is to defend yourself against pathogens and insects. So this is called the metal defense hypothesis. There's a lot of work going on in this area. There are other reasons why people think metal is accumulated in plants on growing on serpentine soils, and we can talk about that during the question and answer session. In California, we do have a serpentine endemic that seems to take up at least 1 to 5% nickel by dry weight, which is Streptanthus polygaloides, which is found in the Sierras. It's in the Brassicaceae. In South Africa, I work with a plant called Berkea cordii that can take up, it's a sunflower family plant that can take up about 10% of nickel. So the idea is that plants take up nickel to defend themselves. But both these plants have specialized insects that are monophagous, meaning that they only feed on this nickelhype accumulating plant. They're specialized plant herbivores that have co-evolved with these high levels of nickel in the tissue. Melanotrichus boydii, named after my colleague and collaborator, Robert Boyd, who was a former UC Davis grad, now at Auburn University, named this bug. The bug is named after him, found only on Streptanthus polygaloides, and this Chrysolina pardalina is also found on these plants. They have different ways that these insects deal with the heavy metals. Some bioaccumulate it, some poop it out, so they don't really accumulate in their body. Understanding this relationship between insects and plants and metal is really, really interesting, even in terms of ecosystem health implications, right? So to wrap things up, serpentine soils are kind of model settings to understand ecology, evolution, conservation, restoration. But unfortunately, these outcrops that are only 1% of the Earth's surface are undergoing a lot of changes due to climate change, development, human encroachment, agriculture, mining. All of these things are threatening. And here is going back to that first slide I started out with, the serpentine outcrop and the sandstone outcrop at Jasper Ridge at Stanford, April 1980s. My grad advisor from University of British Columbia took this photo. Then I took this photo in 2009, this almost the same location, right? But this is kind of what it looks like. There are ups and downs in terms of how much Lascinia is found on this outcrop, but generally Lascinia population has crashed. Native plants are disappearing from serpentine outcrops across the Bay Area. Why is this happening, right? So there's multiple factors playing a role here, but one of the key factors that are responsible for the invasive grasses encroaching serpentine outcrops across the Bay Area and parts of the Mediterranean Europe is nitrogen deposition. There's about 11 to 16 kilograms of nitrogen per hectare raining in the Bay Area, right? So nitrogen that basically is in the atmosphere that falls as plant available forms of nitrogen that has enriched these soils of serpentine outcrops that has now made it possible for the non-native grasses to encroach, thereby outcompeting the serpentine endemic plants. This is a huge problem, right? And what's happening in California, at least in the Bay Area, is like loliums and broma species and goat grasses have encroached the serpentine outcrops all across 280, across 101 in the Bay Area, and they are being basically actively outcompeting the native plants. So this is not only wiping out the serpentine endemic plants or tolerant plants, it also has other levels of impact. Right? So for example, the bay checker spot butterfly mostly has larvae that mostly feeds on Plantago erecta, largely a serpentine tolerant species, and a Castanea species that grows on the serpentine outcrops. The grasses come in, they outcompete these native plants that are food for the larvae of the checker spot butterfly. So there's no food for the checker spot butterfly's larvae. So therefore the checker spot butterfly is going extinct, right? So this is just one of many such case studies where from around the world, where nitrogen deposition and climate change is indirectly impacting the ecosystems surrounding serpentine outcrops. Very quickly, right? Phytomining or agromining is becoming really big across Europe, where people are growing serpentine tolerant plants in old mine tailings and extracting heavy metals from them. I'm not going to show this video, sadly. If anybody's interested in seeing this after we are done, I'm happy to show this short clip of a farmer who used to get rid of this weed for much of his life, is now growing this weed as a crop and basically extracting nickel from the tissue, selling it to a French company and making more money than he has ever made in his life. Right? So basically this whole process is called phytomining or agromining. You use plants that can take up heavy metals, you grow them in the soil, plants take up the heavy metals, you extract the plant tissue, then there's chemically treat the plant tissue, extract the metal that you can then sell as bioavailable metal, as metal ore, right? So this is called phytomining. It's great if you're using native plants in the place where they come from, rather than moving plants around from the Mediterranean to California to do this. That could be a huge problem, as you may have already heard of a plant that was brought to California by a company in the late 90s to phytomine nickel in Southern Oregon, right? But this is in Australia. A colleague of mine, Alan Baker, who's now retired, is holding $70,000 worth of gold in his hand, extracted from Brassica jancea, which is an Indian mustard that is naturalized across the US. It can take up gold when the soil is amended with certain chemicals. It can take up huge amounts of gold that can then be extracted and sold. Berkia cordia in South Africa is grown to phytomine nickel in South Africa, which is great. We shouldn't bring this plant to California to phytomine because it obviously is going to escape and cause all sorts of problems. But agromining is a big, big business right now, especially in Europe, Southern Mediterranean region, and parts of Africa and Australia. But there are some concerns. We've got to be concerned about it. There's a case study of an invasive species in Southern Oregon, Alyssum murale, which became a problem in Southern Oregon and in Northern California. Soil amendments can be toxic to other critters, things we add to the soil to make the metal more bioavailable. We don't want to go the GMO route. We don't want to be genetically modifying poplars and willow trees to take up more heavy metals to sell. That could cause all sorts of problems. You could have bioaccumulation, spread of heavy metals. But I want you to be aware that serpentine plants are now employed actively by humans across the world as a way to mine metal from soil. So if you are interested in all of this stuff that I'm talked about, I encourage you to join the International Society for Serpentine Ecology, where we have conferences every few years. The first conference was in California in 1991, where Art Kruckeberg, Bob Coleman attended. The latest one was in Albania and Greece in 2017, where we saw active agro-mining or phytomining operations in southern Albania. And then all of these conferences lead to conference proceedings that are published with papers that are presented at these conferences. The next conference is going to be in Russia, which was supposed to be last year. Of course, we didn't do it. It's going to be June of 2022. If you're interested in attending this conference, go to the Southern Urals, where there's the world's largest asbestos mine on a serpentine outcrop. No longer a serpentine outcrop, it's an asbestos mine. This is where you want to go. A lot of nickel chromium mines and a lot of endemics as well in the Southern Urals of Russia. So with that, I'm going to end. I just went over a few minutes and I apologize for that. But what I wanted to get across is that these rock outcrops are not wastelands. Like most people think about these serpentine outcrops as all like other types of outcrops, as barren land, wastelands that are not interesting. But these are really model settings to study biological phenomena from the cellular level all the way to the ecosystem level, including having all these really cool applications like phytomining that are now happening on serpentine outcrops that were damaged by mining activities back in the day. If you're interested in the kind of research I do, or if you have children or friends who are looking for geoecological research opportunities, I encourage them to check out my website. I do take grad students. I also work with a lot of undergrads, and I'm happy to end right now. And I apologize again for going a little over, and I'm happy to stick around and answer any questions you may have. Ali, go ahead.
Thank you so much. Thank you. Well, we will take some time for questions. And if you haven't yet typed your question into the chat box, do so now. So the first question, Nishi, brings us back to the beginning. Do you just have a basic general definition of serpentine?
Yeah, I mean, so it's, if you talk to a geologist, they get really annoyed when we call things serpentine, because serpentine technically is a group of minerals that are found in mantle rocks, like things like peridotite, right? So basically, ultramafic, there's a question and answer, one question that talks about ultramafic. Ultra means high, mafic means high iron and magnesium. So any type of rock that have high levels of iron and magnesium is lumped into that broad umbrella called ultramafic rocks. Serpentinite is just one of many, right? So in European literature, people use ultramafic because they are talking more broadly about all of these different type of rocks. In California, we have a lot of serpentinite. So we generally call everything serpentine, which Europeans don't like. Like every time we go to this conference, they're like, you don't even know whether it's serpentinite. It could be peridotite. It could be, some other type of ultramafic rock. But because they have that kind of serpentine type mineral in it, we just call them loosely serpentine. So I've started leaning towards calling everything ultramafic, unless I have a geologist who've done a mineralogy study of the rock and tell me whether it's peridotite, lizardite, serpentinite. It tells me what the real rock is, rather than not knowing what it is and just kind of randomly calling it serpentinite.
That clarifies it for me, and I hope for everyone else too. Thank you. And you also answered that question about ultramafic rock too. That's the umbrella. Let's see here. Alyssa asked, are the metals more available when you were speaking about the nickel? Are metals more available at a higher or lower pH? And she was wondering if the pH affects availability.
Totally.
Where you find.
Excellent question. I mean, I wish my students asked these kinds of questions. Yeah, excellent question. pH dominates ion availability, not just heavy metals. Everything's availability in the soil, ionic solubility and availability is determined by pH. For heavy metals like nickel, generally lower pH is better, right? So serpentine soils are generally neutral or higher pH. In those settings, heavy metals are not as bioavailable, right? When the pH drops, they can become more soluble and become more bioavailable. So every ion in the periodic table that's in the soil will have a particular range of pH that makes it more or less soluble, right? And then if the plants are experiencing that pH range, and pH is changing all the time, it rains, there's a bunch of organic matter that gets dumped in the soil, pH is constantly changing, and there are range, I mean, it's not an exact number, but it's a range. So when it falls into that range, if the nickel becomes bioavailable, and if the plant is able to take it up, then it could potentially get in. Nickel can get in through zinc uptake systems, iron uptake systems, calcium and magnesium uptake systems. So it basically creeps in. There's not a specific nickel uptake protein, but nickel looks like calcium and magnesium. It's about the same size. So it can kind of creep in with other ions on the transporters that the plants are taking other ions in, because nickel is really high, right? And those other ions that the plant is trying to get is low. So there's ion competition happening in the soil solution, which basically drives some of these toxic elements into the plant.
Thank you. And that's one of the biggest threats to these ultramafic, these serpentine outcrop landscapes is nickel mining companies wanting to extract and these natural resources that we're trying to protect.
Yeah, I mean, so that's the thing. Like, I mean, I don't know whether you're familiar with that little video link. I mean, if there is a way for me to send those video links to you, one of the problems, there is a video link that I think that was done by the some Siskiyou organization of Alyssa Murale, that was brought into Southern Oregon by a company called phyto something. I can't remember the exact name, but they basically brought it in and they were mining nickel in some place in Southern Oregon. And this has happened in the 1990s, and they had promised that they would weed this crop after the mining operations ended. But the company just disappeared and the plants escaped, right? And I remember sitting in an audience in Cuba when this was discussed, or maybe even in South Africa a long time ago. And I was just a young student, and I raised my hand and I said, you're bringing a Mediterranean plant to another Mediterranean part of the world, and it's the serpentine endemic. So what are the chances of this guy going and escaping onto serpentine outcrops in California, right? And people just kind of didn't even listen to my question. But of course, that's what happened.
We're still dealing with it
Yeah,
here in Southern Oregon.
Exactly, right? I remember asking this question. It's probably like one of the first questions I asked in a conference, because I felt like, I mean, when you're a younger student, you don't usually ask questions from big researchers. You're like little, you're afraid to ask questions. Little shy. And for me, this was like such a, I'm like, this is bound to happen. So I've got to ask this question. But of course, they brought it in, the company disappeared. And there's also patents for some of these plants now that are by the USDA and some private companies. They patented the nickel accumulating behavior, I guess. So they are the only ones that can use this plant to restore landscapes and things like that. I mean, it's problematic, like all patents are.
Yes, and I don't know if you're aware, but we have a bill on the table right now called the Southern Oregon Watershed and Salmon Protection Act, which will permanently protect a lot of headwaters here in Southern Oregon, where a lot of these serpentine outcrops are located, which have those nickel laterite deposits that we're trying to protect. So for all of you listening that are still here, go to the KS Wild website and learn more about it, and write Senator Ron Wyden. Thank you for introducing that bill. We hope it passes.
That's so great. And I just saw a comment from someone called Stacy. I mean, I would love to get some photos of, I don't have photos of Addison murale that I've taken from Oregon. If you guys can email me any photos you have of plants that have like taken over your backyard or serpentine outcrops, please email them to me, because I can use them in talks, because I use it as a model to show why we should move plants around for green technologies. So Stacy, I would love it, and I would really appreciate it. Drop me an email, and I'll give you credit, and I'll use your photo when I give talks elsewhere. It's a huge problem that is, California was the first place this happened, but it is going to happen in other places, because people are moving plants from one serpentine outcrop in one country to another country for these so-called green technologies. And that can do more harm than good.
Yes, and thank you, Stacy. I'll make sure to include your email in the follow-up email that will go out here tomorrow.
Yeah, I can also just type it in for people who are interested.
Oh, there you go. And with that, that is our last question. And that's a good place to end, I think, for tonight, unless you have anything else you would like to add, Nishi. Now is your time.
Yeah, I mean, I already kept you guys. I mean, I can talk about this for as long as you want, but I also am aware of the fact that it's dinner time and people need to get out. So we can end here. And if you want to continue this conversation via email, and I'm also available to chat with people on Zoom if they are interested in talking about any specific topic that I discussed. So all they have to do is send me an email, and I'm happy to get online and talk to them, and hopefully see them in person next year.
I hope so.
I would love to visit the Klamaths. I just have not been there in recent times. So we'll try to do it at some point in the future.
Well, we would love that. We would love to have you come visit. So let us know, connect with us. And thank you again for your time and your expertise and your knowledge. Thank you so much. I so enjoyed tonight, and thank you to all the attendees. And with that, have a great night, everyone. Thank you.
Have a good night, everybody. And Ali, thanks so much for the invitation. I appreciate it.
Thank you so much.
Bye.
Bye.